Hi, I'm Keay Nakae, one of the senior healthcare analysts here at Chardon. I want to introduce our next guest from Univercells Technologies, Chief Technology Officer, Tania Pereira Chilima. The format for this session is roughly a 30 to 40 minute fireside chat. If any of our audience attendees have questions during the session, feel free to type them into the question box under the video player, and we will try to ask them on your behalf. Tania, welcome.
Thank you very much, Keay, and thanks, Chardon, for the opportunity to speak in this event. So I'll start by introducing our company. So I am part of the Univercells group, and Univercells is a Belgium-based company focusing on making biologics available and affordable to all. This vision has been, or it's been achieved through the different subsidiaries that Univercells Group incorporates. There are 4 of us. Today I'm here, so I'm the Chief Technical Officer of Univercells Technologies, so it's one that I represent, and we are really focusing on revolutionizing biomanufacturing through technology innovation. There's also Exotera, who is a CDMO with a focus on gene therapies, and this company has a high affinity for best-in-class processes and associated technologies to provide significant benefits to the customers. So Univercells Group was founded by a couple of serial entrepreneurs with a lot of experience in biopharma and bioprocessing. Here you see José and Hugues, and José was actually at the inception of the Untitled bioreactor, which is now currently being commercialized by Keay Life Sciences. Another example of a success story here is the MasterCell group, MasterCell that was recently acquired by Catalan as well, so they were also involved in that. So, as I mentioned, I represent Univercells Technologies. We are a company with over 90 employees, and although we are based in Belgium, we have a global reach, and we are supported by a number of different distributors across the world, which enables us to basically commercialize, install, train our customers, and support them in the adoption of our system. So this slide just summarizes some of our key customers, as well as some of the funding bodies that have believed and invested in Univercells and Univercells Technologies. So looking at customers first, there are some names here that I'm sure that you can flag, especially in the gene therapy space. So the name of, for example, 4D Molecular and also KCT. When looking at the funding bodies that we have welcomed, so we have some funding from the Bill and Melinda Gates Foundation that really has enabled us to accelerate the development of our technologies, as well as more equity funding, such as private equity funding, such as KKR, in the framework of Gamma Bio Life Science. So now looking at switching a little bit to some of the technology challenges around the gene therapy space. So this slide just shows an extract of products in clinical trials for different indications. And what you can see here is that when we look at the prevalence of these indications versus the predicted dose sizes for these drugs that are currently under development, the combination of both results in a significant range in annual demands in terms of viral particles needed per year to treat the different patient cohorts associated with the different indications. And this particular slide shows this landscape for AAV-based gene therapies, but the slide is it's quite interesting because it really shows the need for technology flexibility in order to enable companies to be able to manufacture the products with very low annual demand, like ophthalmology applications and so on, and also the kind of muscular dystrophies and so on, which have the highest end which are at the higher end in terms of annual demand. So then, when we look at the current technology landscape, what we see is that technologies that today there are several technologies available in the market for gene therapy manufacture, and this can range from flatware, so you can have cell factories, hyperstacks, and also have packed bed bioreactors. So this could be and also a more scalable alternative to flatware. But you can also have certain bioreactors operating in suspension. And when we look at the target scale, if we are looking at very, very, very low scale applications, it is possible to still remain in flatware. When we go to medium scale applications, depending on the type of cells that you're using, whether you're on suspension or adherent, you might have to use. So if you're in suspension, you would go for a certain bioreactor. If you're in adherence, you go for a packed bed bioreactor. But we see here already the expression system used dictating the technology that is to be adopted. If we're on the higher end, so if you really, really need to go to a very, very, very large scale application, what we see is that customers or the industry is moving towards suspension in order to achieve high scale. And that, of course, requires the cells that are adapted to suspension. So there's currently no solution for adherent-based cell manufacture or using adherent-based adherent-based viral vector manufacture at very, very high scale. So we see here the kind of technology limitation where if you're at the very, very high end of the annual demand requirements, then you have to go into certain suspension. What's also important to consider about adopting when adopting these kind of large bioreactors, certain bioreactors of thousands of liters operating in suspension is the footprint requirements. So if by any chance there are capacity somehow facility constraints and it cannot accommodate the suspension bioreactor and all of the tanks that you need to move the volumes from one unit operation to the other, there's also something to be considered during technology adoption. So Univercells has developed a technology portfolio that would basically enable us to overcome this challenge of being limited by the technologies when you want to go to scale or whether you want to use suspension versus adherent cells. And what we have basically is a range of different technologies available in different configurations. So you can go from 10 meters squared to 600 meters squared manufacture in our systems. And that basically allows you to kind of cater for different dose indications with different annual demands using the same technology. This technology has demonstrated, or we have evidence with different customers that this technology has enabled different, so higher productivity with respect to an alternative technologies. So when we move to higher annual demands indications for those who don't want to move towards suspension cells, they can remain in adherence while still achieving the high productivity, which would basically enable them to cater for their annual demands. And I will show some results of what we have achieved in partnership with our customers with our systems versus competing technologies. Ease of process transfer. So an additional feature of our systems that we have evidence is that we were able to transfer both adherent and suspension systems with a limited number of runs from the technologies that were the reference process, using reference technologies, which were cell factories and shaken flasks for both adherent and suspension, respectively. So we were able to transfer those processes into our systems with minimal amount of process optimization. Also process flexibility. So I've been talking a lot about suspension and adherence. Actually, this technology enables you to use both suspension processes and adherent processes in the same technology. So this means that for example, for CDMOs, if you have a room or a single room which you need to utilize, you can imagine to have the same system which is scalable, and you can have up to 600 meters squared within a very, very small volume, and you can have the same system and you can use it for both customers using adherent processes, others and customers using suspension processes. And this, I believe, there is a unique opportunity to basically reduce, to basically increase the utilization of your suite. For gene therapy innovators, this is also an opportunity to not to have to adopt different technologies for different applications, if that makes for different applications. So if you have a portfolio of different products coming down through clinical trials, you don't have to basically adopt an adherent-based process and later go to a suspension-based process, because you realize that you need higher scale to tailor to the needs of one versus the other. And last but not least is the footprint of these systems. So our technologies are designed to basically they are designed on the base of using the principles of intensification and chaining, and this basically enables us to have very, very high densities in a structure fixed bed design, so very, very high cell densities in a structure fixed bed bioreactor. So this is how intensification comes about, and this already reduces the operational footprint of the process. And in addition to that, we link this bioreactor with midstream operations, so with clarification, concentration, and PS and diafiltration, and this further reduces the operational footprint, because all of a sudden now we are basically operating in this integrated and continuous mode, which means that we are reducing the size and the number of hold tanks, basically. And this is basically what you see here, and this system it's the NevoLine system, which embodies these principles of process intensification and tuning to significantly reduce footprint and to basically incorporate different unit operations within a very, very small module, basically. And this is our technology portfolio, and you've seen the NevoLine from the previous slide, but it all starts at the smaller scale. So the hydro bioreactor, this is our proof of concept system, which enables customers to rapidly adhere to the technology. And then we move to our middle-sized system, which is one of our flagship products, which is the ScaleX Carbo bioreactor. This system is available in both 10 meters squared and 30 meters squared configuration, and can be used for clinical trial manufacture, and for certain applications, it will be the final manufacturing scale as well. Finally, we move to the nitro bioreactor, which is the highest scale system, and this system is basically here at the bottom of the NevoLine. So it's integrated into the NevoLine, and this system is available in both 200 meters squared and 600 meters squared configurations. And together with the NevoLine, they really enable process intensification and chaining to reduce the overall operational footprint. So now with this technology portfolio, what we are achieving is that we are able now to decouple the cell type used from the technology. So you can use suspension or adherent cells within the same technology. We are able to have a large scale system, so the nitro is available up to 200 to 600 meters squared. Not only that, it's also available in smaller configurations to enable that flexibility that I was mentioning to accommodate products with very different annual demands. And on top of it, we are also having a system that has very high performance. As I mentioned, we have evidence that with the ScaleX bioreactor, we are able to achieve higher productivity than with traditional technologies. And finally, these systems are also designed to reduce manufacturing footprint. And I just mentioned to you that the ScaleX has shown to provide higher productivity with respect to competing technologies. And this slide is just a snapshot of some of the results that we have gathered with our some of our key customers. So starting with the AAV case study, this was done with 4D for DMT, so 4D molecular therapeutics. And in this case study, you can see basically the 4 runs performed with the ScaleX bioreactor, and we were able to achieve comparable results with respect to the reference process in cell factories. And these results were 4 times higher than traditional packed bed systems. Looking at also a study performed by KCT. This one is on lentivirus, and what they've seen is that with the ScaleX bioreactor, we were able to go higher than double the productivity per square centimeter of the bioreactor. And now switching to suspension cells, this case study was done with Pereira. Again, for this one was an adenovirus-based process, and the comparison was done against shake and flask. And this was just 3 runs in the ScaleX to look at whether we could transfer this process from the shake flask to the ScaleX bioreactor and to see whether the cells were going to entrap and whether there was going to be an impact on productivity of having these suspension cells entrapped in a structure fixed bed bioreactor. And what we saw there is that consistently across the 3 runs, the results in the ScaleX bioreactor were superior than those of the shake flask. And this is without any significant or any really process optimization. So we're really excited to see what will happen, as we see more and more of these case studies, and we help our customers to perform more and more process optimization around suspension cell cultures trapped in fixed bed bioreactors. Maybe just one final slide on the technology readiness or the adoption of our systems. So this year we'll see around 90 ScaleX and ScaleX controllers installed across the globe, and we will see as well around 6 NevoLines being installed as well. The majority of our customers are in the gene therapy sector as well as on the vaccine sector, even though we also see customers using our technology for other applications such as exosomes, exosomes or even proteins. And the majority of our customers and our revenue is split between EU and the US. Thanks a lot for your attention, and I welcome your questions.
Great, thank you. So Tania, a number of our presenters have talked about, and when asked about future areas of improvement have cited intensification and chaining as parts of the manufacturing process where there could be benefits derived. So can you talk about those and how your technology is advancing those two characteristics?
Sure. I put a slide for that. So indeed, so when we look at standalone unit operations, you can see that there are, when you look at traditional processes, they operate in using standalone unit operations, and you basically will have tanks, intermediate tanks connecting them together and taking the product from one operation to the other. This batch processing has been done and it works, but it does result in very high operational footprints. So what we are doing is to basically have high capacity at small footprint. And to do that, we use these principles of intensification and training to enable integrated continuous manufacture in order to really remove these intermediate holding steps and also basically synchronize doing different unit operations together so that you can basically also reduce the final duration of the midstream steps, because you can imagine that what we are doing is that we are linking the virus production with virus classification and virus concentration. So this means that the virus can be clarified and concentration and concentrated as it's being produced. And at the end, you have this concentrated bulk and clarified that can be easily transferred to downstream. So this is the concept of chaining. The concept of intensification, it starts really in the bioreactor. We have a structured fixed-bed bioreactor which has a lot of which basically enables a very, very high densities to be achieved. As if I take the example of our largest bioreactor, the Scalex Nitro, it has 600 meters squared within 60 liters. So the bioreactor itself is very, very, very small. So you can imagine that, for example, if you have an intracellular product and your cells are all contained within 60 liters, it means that at the end of the process, you only have to take to treat and lyse 60 liters worth of cell culture, because the media is decoupled from the bioreactor. This, of course, has several benefits because the volumes that you have to treat subsequently are a lot lower. And also the reagents that you have to use, so the volume of the lysis reagent, if you use detergent-based lysis steps, will also be a lot lower. So those two together have not only operational, footprint reductions, which would result in capex and opex, capex and opex reductions, but also they reduce the amount of or could, depending on the process, they also could reduce the amount of key reagents used.
Right. Great. Another important advancement of yours, the ScaleX bioreactor platform, is its application for both suspension cell culture as well as adherent cell culture. So, especially, with respect to your, CDMO customers, what are the implications of having a dual use platform, especially, for cell and gene therapy?
Well, the CDMO, as I mentioned, I think is a unique opportunity to really increase capacity utilization. So you have a whole platform that can be used for both suspension and adherence, so that you don't have to have a room or equipment that is idle because the customer has a process that is not suited for that particular equipment. There are, of course, many other benefits in general of having a structured fixed bed bioreactor or a fixed bed bioreactor that can be used for suspension cells. One of them is ease of process intensification. So in the same way that with adherent cells, we have the cells the cells basically staying in the bioreactor while the media flows in and out. You can do the same thing now with suspension cells, where the cells are basically entrapped in the bioreactor, and you can flow the media in and out. And this really eases the adoption of perfusion processes because you don't need an external, cell retention system, which sometimes can prove to be detrimental to cells. In addition to that, is that with our platform, we are also facilitating integrated continuous processing, so we can have link the bioreactor straight with a clarification sequence and a PFS sequence, as I mentioned, for both adherent and suspension cells. For suspension cells, given that you can have them in a wave bag and you can expand them in the wave bag, this means that if you have a process that has suspension cells and you can put them in wave bags during the seed train and have a very high cell density in the wave bag and then inoculate that into a high cell density bioreactor, you're really debottlenecking the seed train, and now you have an overall intensified upstream to midstream process. So you have the pre-culture being intensified as well as the cell culture and bioproduction stage. Another one would be the ease of development. So as I mentioned, we have successfully transferred both adherent and suspension processes to our technologies with a minimal amount of process runs. So that could really help accelerate process development. And given that our technologies are developed basically maintaining the same scale-up parameters across the scales, this system can also ease the scale-up process across the different scales and throughout the customer journey. I've also mentioned already the benefit of having an enhanced performance with respect to traditional technologies, which means that we can achieve higher scales without having to go to a stir tank bioreactor or without necessarily having to scale up to a big extent. One thing that is also worth mentioning is that the fixed bed itself behaves as a filter. So at the end of the process, a lot of the cell debris and the cells can be retained into the fixed bed. So even with suspension cells, this can be retained into the fixed bed while the virus can be collected, and that can also streamline the downstream process. And yeah, I think I could go on, but perhaps we can go to another question.
Yeah, I think that clearly your innovation is well thought out to minimize the adoption risk. One of the again, your key piece of equipment, the Scale X, is competing with the iCELLis, which seen very good acceptance into the market. Can you compare and contrast Scale X with iCELLis and why you believe it's better?
Yes, I can. So in my presentation, I've shown a couple of results that demonstrated the performance of our system versus iCELLis, so using an independent group. So indeed, we believe that the benefits of our system is really down to the to the structured fixed bed design. So this structured fixed bed design basically enables an even cell distribution, and during inoculation, the cells evenly distribute across the fixed bed. And we actually have performed experiments when we dismantled the fixed bed and measured the cell density at different points within the fixed bed to then look at how they differ or not. And indeed, we found that the cell density across the fixed bed was quite homogeneous. But this also means that there is a homogeneous kind of access to nutrients during cell growth and virus production, as well as access to virus or transfection needs. So we really, and we don't have kind of this concern of having, potentially preferential pathways where you have a packed bed system, where maybe some cells are more exposed to nutrients than others, and so to fresh nutrients than others, and so on. And we believe that this is the cause of this higher productivity with respect to the isolated bioreactor. But of course, we are still learning and learning more and more by seeing more and more processes coming with this kind of productivities with in comparisons with the isolated bioreactor.
With the COVID pandemic, a lot of single-use supply chains have been severely affected. How does your technology, ScaleX or NevoLine, reduce supply chain risk?
Yeah, so they're by design integrated and continuous manufacture. So our technology, what it does is that it really reduces the number of components that are needed for the process. So we reduce the number of manifolds. We reduce the size of the different process components. For example, CSS. If we are concentrating throughout the process, it means that you don't have to concentrate a large volume at the end of the process, and we can potentially downsize the size of the cartridge. And last but not least, if we consider, for example, the number of whole tanks that you need across the different steps of a traditional process relying on standalone technologies, what you'll see is that by removing that, we are also removing the number of bags that you need. So you don't have to so it's basically a reduced strain on your supply chain, because as we all know, there have been a lot of challenges in getting access to single-use systems such as bags.
And what level of involvement has Univercells or your equipment had in the development in response to COVID-19?
Yeah, so I think there is a very nice slide that I could use for that. So this is just an example of some of the projects that we are involved in. This one it's basically a recent project that we are collaborating with. So we recently signed an LOI for two different collaborations. One of them is with the Institute of Health Research, Epidemiological Surveillance and Training, and the other is with the Institute Pasteur, both located in Dakar. And the first one, it's basically tailored at or it's targeted at working together to basically create a bioproduction capacity of vaccines for immune immobilization. But the second one, which is more relevant to your question, this one is this collaboration with IPD, it's aimed at establishing local production of COVID-19 vaccines. So this is an example of how our systems are being used, and how Univercells and Univercell technologies are being involved in such initiatives.
And so, when you have innovative technology, what are you finding are the primary obstacles in trying to get large pharma, biotech companies, or CDMOs to adopt your technology, and how might COVID-19 and the response to that accelerate the adoption of continuous manufacturing processes that you support?
So to the first part of the question, so there are different reasons. So one of them certainly being companies that have legacy facilities, so they have existing installed capacity, and they might have PFS systems, qualification kits, and they want another bioreactor to incorporate into that. The NevoLine is really an integrated platform, and it includes different operations together. So to now to basically change or to adopt a new technology that will replace some of the existing systems that are in place can be challenging, and it can it will need a lot of a good rationale from the company to their stakeholders, and that's something that it's one of the challenges. Another one is conservatism. So companies that are that they have their processes and they know how to operate in batch mode, and that's also changing the way of thinking, and thinking more to the benefits of have them understand or fully understand the benefits of integrated continuous manufacturing, and how it can benefit their day-to-day operations is also one of them. Another one is, of course, this is a new technology, and as that, I think a lot of companies would prefer to see other companies go through, registering products with this technology before they go first and they adopt the system. So yeah, it's a matter of changing the way that we see biomanufacturing, and that does take time, and does take understanding and explaining, and also generating data that emphasizes the benefits of adopting such technology principles.
Yeah, so I guess to that point, one of the questions we've received from the audience is, have any of your bioreactors been used to make clinical AAV material that you're aware of?
Not clinical AAV material, but it has been used to make clinical material for other applications, not AAV at the moment.
Okay. Okay, well, we've reached the end of our time here. I really want to thank you, Tania, for joining us today. And with that, have a good rest of your day.
Thank you. Bye.
Yeah, thank you.